All right, thank you very much. I'm really happy to tell you all what we're doing at ILA Pharmaceuticals. It's going to be a really exciting next few months for us. If we can go to the third slide, please. So at ILA Pharmaceuticals, at its core, we are a mid-clinical stage drug development company. We're focused on repurposing small molecules for the rapid development of antiviral therapeutics for infectious diseases. Next slide, please. I'm excited to tell you today about our lead program. It's called ISLA 101. We're developing ISLA 101 as a preventative initially for dengue infections. We're moving forward with a phase 2 clinical trial that we expect to start early next year. We call this the PEACH clinical trial, and I'll tell you more about that as we go through the presentation. You may ask why we're focused on dengue. Dengue is a huge problem. There's 390 million dengue infections a year. There's no antivirals that work against it. The vaccine possibilities are really small right now. And so there's a significant unmet medical need, which means there's a significant market opportunity for us. The program is supported by phenomenal data, both at the bench, looking at cell culture data, also in animal models. We have robust animal models of dengue infections where ISLA 101 is proven to be effective and protective. As a result of all of this, we have some really neat commercial opportunities, one of which is the potential for a priority review voucher. I'll tell you about that throughout the presentation. And while ISLA 101 is our main focus, we are looking at other alternatives, pipeline expansion strategies that we're working on right now. Next slide. So I mentioned that we're a repurposing company, and you may ask, what does that mean? Well, to develop a drug from start to finish typically takes about 10 to 15 years. It costs about $2 billion, if not more. It's a really long and expensive process. And that is exactly what we do not want to do. What we want to do with repurposing is identify a molecule that has clinical history in one indication. It's got a good safety profile. And then we want to use that data to use the molecule in a different indication. So for instance, our lead molecule was initially developed as a cancer therapeutic. There's a lot of data associated with that molecule demonstrating that it's got a good safety profile. We're going to use it as an antiviral molecule. The beauty of doing this is that we're able to move straight to a phase 2 clinical trial. And for those of you who look at drug development stories, when you get phase 2 proof of concept data, that's generally a really significant value inflection point. So for us, this is a rapid way to get to a really valuable inflection point. Next slide, please. So the challenge, next slide. Yeah, thank you for this one. The World Health Organization recently came out and said dengue fever is the most rapidly spreading mosquito-borne virus infection in the world. That's a really profound statement and highlights a significant problem. Next slide, please. We've taken some articles from really around the globe over the last few months just to highlight the fact that dengue is a huge problem. We have articles from Australia, the UK, Nepal, the United States, Singapore. What we're seeing is that dengue is cropping up over and over again in places, in fact, frequently places we haven't seen it before. For instance, France or Miami are having infections and outbreaks of dengue right now. The thought is that as a result of global warming, we're seeing places be warmer than they've been historically. We're seeing changes in weather patterns where places are wetter, there's more rain than there's been typically. And what that means is the mosquitoes are following the weather. The viruses are coming with the mosquitoes, causing these outbreaks in places where we haven't seen it before. So a significant unmet need. Next slide, please. So, why dengue is the first target for ISLA 101. ISLA 101 has activity against a variety of mosquito-borne viruses, but just the numbers are staggering. Half the world's population is at risk of an infection with a dengue virus. We're seeing increasing spread of the virus to the US, to Europe, to Australia. The good news is that ISLA 101 preclinically has activity as both a prophylactic molecule and a therapeutic molecule. We're moving forward initially as a prophylactic, so we want to see if we can prevent the infection. We could in the future pursue a therapeutic strategy as well. Again, the program is supported with phenomenal preclinical data. We're excited to be able to move forward with dengue initially. Then upon approval there, we can springboard to other mosquito-borne viruses. We could do expansion studies looking at Zika or West Nile or Japanese encephalitis. Next slide, please. So what is ISLA 101? I gave you a preview a bit ago. ISLA 101 was initially developed as a cancer therapeutic many, many years ago. It's never been approved anywhere for anything. And it's interesting because the molecule has been in 40 plus clinical trials. It's been in literally thousands of people in many clinical trials around the world, but never approved. Not because of safety reasons. It's got a really good safety profile. It just hasn't been demonstrated to be an effective cancer therapeutic. So what this means is that regulatory agencies around the world have already seen the molecule. They've already approved clinical trials being initiated. We are able to rely on all of those data from other people and move to a phase 2 clinical trial. Next slide, please. I mentioned the preclinical data package that we have, and again, it's really phenomenally robust. With our collaborators at Monash University, they were able to identify ISLA 101 as a molecule that prevented infection of all 4 strains of dengue, as well as Zika and West Nile. We performed the yellow fever studies. In addition to that, they took freshly isolated human cells and checked to see if ISLA 101 could prevent infection of human cells. These are the cells that would be infected if you were to get an infection. And in fact, ISLA 101 was protective there. Furthermore, I mentioned previously, we have really aggressive animal models of dengue infections as well as Zika infections, where ISLA 101 protected against those infections. So with the safety data from other people, with this preclinical package, we went to the FDA and had a pre-IND meeting with the FDA and laid out our plans, asking them if they agreed that we could move to a phase 2 clinical study. And they agreed this was the right approach and we had sufficient data to move forward. Next slide. So what do we need to do to get into the clinic? We need to make our drug. We need to finalize the investigational new drug application, or IND. And we need to finish the clinical protocol that's going to describe what's going to be done in the clinical trial. So I can tell you that the protocol is complete. I can tell you that the manufacturing of the clinical product was complete a few weeks ago. We now have drug made. That drug is sitting in stability chambers right now. We have to get some analytical data out of the manufacturing run, but that's the last piece of our IND that we need. Once we get the manufacturing data, we can finalize the investigational new drug application. We're expecting to get that data maybe the first week of December or so. We expect to file the IND by the middle of December. Once you file the IND, there's a mandatory 30-day period that the FDA has to review your program. If you don't hear anything from the FDA, you're good to go. You get to start clinical trials. So again, we expect to be filing our IND mid-December. Hopefully we have a cleared IND by the middle of January. Next slide, please. So I've got to show you this. I mentioned that we just had our drug made a couple of weeks ago. Here is that product. So on the left, these are our collaborators who manufactured the drug. They've made it. They're putting it on these trays so that the products can dry. In the middle, you can see my blue gloved hand holding the capsules that we'll be using in the clinical studies. And then on the far right, these packages or these capsules were bottled and they were then put in these boxes where they're awaiting a clearance to be shipped to our clinical site. So I'm actually really incredibly excited about this. This was a really phenomenal piece of work. So next slide. So once we get the drug to the clinical site, what are we going to do with it? And the study that we're going to run is called a PEACH study. It stands for prophylactic examination of an antiviral in a dengue challenge model. And we know that that's a mouthful, so we're going to call it the PEACH clinical trial. It's a very slick study. What we're doing, it's a challenge study. And this means that we enroll healthy individuals, and then we infect them with an attenuated or weakened strain of the dengue virus. People really do sign up for this. They get paid to be in this study. But what it enables you to do is follow the viral life cycle in these patients, because they're in the hospital almost every day throughout the course of the first couple of weeks of the study. We can take blood samples, we can monitor how severe the infection is, et cetera. So fortunately for us, Walter Reed, the Army Medical Hospital in the United States, in collaboration with SUNY Upstate in Syracuse, New York, which is where we're doing this study, they recently completed a phase 1 clinical trial using the same virus that we're going to use in our clinical trial. And what they wanted to do was just study the infection, see how people responded. And what we learned was that people get a little sick. they feel a little bit crummy, they get a little bit of fever, but nobody gets very sick. And so we recognize this is a perfect model for us to go in and try to block the infection in a hospital setting in a well-controlled study. So we will enroll healthy individuals, pre-treat them for 3 days with ISLA 101, and then infect them and see if we can prevent the infection. While there's lots of things we'll measure in the clinical trial, we're really focused on, can we prevent the infection? Next slide, please. So the commercial opportunity, there's a few ways this is going to go. You might think, because we're looking at a virus that's in certain endemic regions where people may travel to these regions, that this looks like the traveler's market, maybe thinking analogous to antimalarials, and that would not be wrong. We think that there's a really good analogy with antimalarials, and in fact, that's about $1 billion market right now. So we think that that's a fine thing. We also think the military is going to be interested in what we're doing. We have a CRADA, Cooperative Research and Development Agreement, in place with the Army. Under that agreement, they're giving us the virus that we're using in this study. They're also giving us the data out of that study so that we can use that as a comparator arm. So they're well aware of what we're doing. We also know that dengue pops up in Brazil, in India, and they have enormous outbreaks. And we think that there's an opportunity with these governments to potentially stockpile ISLA 101 to curtail these outbreaks when they happen. Finally, I'll mention the priority review voucher. This is a really unique opportunity. And these vouchers are incentives that were created by the FDA to encourage companies to develop drugs for underserved diseases. And the way it works is if you get your drug first approved on an indication on a list of eligible diseases, then you get this voucher. And the voucher enables you to get a second program, the review expedited at the FDA. So not your first program, you get this approved with a voucher, and the voucher enables you to work on a second program and get priority review. And the beautiful thing about the vouchers is you can sell them, and Big Pharma really likes to get them, and they typically sell for about $100 to $110 million. So while our goal is to get the drug approved and launch a product, the priority review voucher is a really nice icing on the cake if it's available when we get there. Next slide, please. We do want to advance our pipeline. I won't spend too much time on this. We've identified some characteristics of other viruses that we're very interested in, and there's some exemplary viruses here that we're looking at. These will form the basis of some work that we're doing with our collaborators at Monash University and Griffith University. Next slide, please. So where do we go from here? Over the rest of this calendar year, you should expect us to see that we're going to get the clinical material released. We'll have the stability data. We're going to get that material released. We're going to file our investigational new drug application. These will be the primary events. Then as we roll into January, we're hoping to see an open investigational new drug application, so open IND. This will enable us to start screening and enrolling and dosing people in the PEACH clinical trial. We'll advance through the different cohorts. We're doing 4 cohorts of 4 individuals. Each of the cohorts is 2 to 3 months, so it's actually going to go fairly quickly once we get into the different cohorts. And then, of course, we'll be looking to identify lead molecules out of our research collaborations. Next slide, please. Just want to highlight, we've got a phenomenal team from, management and board and our collaborators, our scientific advisory board, just top-notch team. Very excited to be working with all these folks. And so I think the next slide's my last one, and I'll leave it right there. Thank you very much.
Thanks, David. That was fascinating, really interesting. Just when you had that slide on the board and management, in looking through your top shareholders, there's Bill Garner has got 27%, and Kessler Partners 13%, and Jason Carroll, they own around 50% or so. Who are they, given they're not on the board, and what's their involvement?
Yeah, so Bill Garner he was one of the founders of ISLA Pharmaceuticals, the predecessor to Island Pharmaceuticals. So he and I've worked together for a number of years. Bill stays close to the company. Obviously, he knows what we're doing, and we communicate with him and keep him in the loop with things, but he's not formally associated with the company. Kessler Partners is actually with Al Hansen. He is on the board, so we do have the shareholder there. Jason Carroll is a major shareholder we're really grateful for. He's incredibly supportive of the company. Obviously, he's not on the board, but we do keep in touch with him a lot, just to make sure he stays apprised of what's going on.
Sure. And just going back another step, when you repurpose a drug, who owns the patent rights? How does that work?
Yeah, that's a great question. So it depends. There are instances when somebody actually does own the underlying composition of matter, and if you were to repurpose that drug, you would have to deal with them, right? In this case, ISLA 101, the active ingredient of ISLA 101 is off patent. It was developed really long time ago, so nobody owns the underlying composition. Now, what we've been able to do is license technology from Monash University that relates to the use of ISLA 101 for the prevention and treatment of mosquito-borne viruses. So that's one way that you can capitalize on a repurposed asset, is getting method of use type claims. Other times you can get different formulation claims. There's ways to protect it, but in our situation, the core composition, it's off patent.
Yeah, right. I see. And so when you take ownership effectively of that drug, how did you end up matching it with dengue? Is it driven by the data, or is it driven by the opportunity?
It is 100% driven by the data. So we were looking for molecules that would have an activity against mosquito-borne viruses, Zika and dengue. And we've identified a publication, Bill Garner actually identified a publication from David James at Monash University, where he, in his lab, did screens looking for molecules that would prevent the infection. He screened a library of molecules. It was a very unbiased, very scientific approach to find a molecule that would be effective. And in fact, what he pulled out was ISLA 101, or the active ingredient in ISLA 101. And then only then did he discover this is a molecule that we know a lot about. This molecule's been around for a long time. It's got a great safety profile. So the conclusion of his paper is that ISLA 101 ought to be developed as an antiviral for mosquito-borne viruses. So, we found the publication. We were able to get the license from Monash University, and then we're developing this, and looking forward to being in the clinic very soon.
And of course, it's been a really tough time for biotechs in general. How's this affected Ireland?
So where we have been affected, we've had some delays. Admittedly, we've had some delays. Things, believe it or not, COVID really affected supply chain. We had trouble getting some reagents to get some things made, to get some of our ingredients made. So, that has slowed us down. It has been a tough time, but we've also found some really good partners along the way. And I'm happy to say that they have been phenomenal to work with. And I think we've really rounded a corner now that we have drug literally in our hand, and moving it all forward now.
Yeah, literally in your hand.
Right.
And is so the next catalyst, is it more positive data? Is that going to drive a re-rating? Where's the next steps?
Yeah. So what I think is going to happen is, as we start moving to the clinic, I anticipate there's going to be anticipation and optimism for the company and the program and the share price, right? I think one of the next pieces of news will be when we file the IND. I think that will be a significant piece of news for people to be watching. When we get the IND open, which again, we're expecting that to be in mid-January, that should be a really big piece of news, because it means we've got the green light to get into the clinical trials. And then it's a matter of enrolling and dosing and seeing how it plays out. But I think that we're going to see significant increase in interest as we move towards clinical trials and getting into the study.
Thanks, David. That's all we have time for. Really interesting presentation. Thanks for your time. Have a nice weekend.
Thank you so much. Yeah, much appreciated. Bye now.